It's Dr. Anatolij Horuzsko, and I'm working in Georgia Cancer Center.
My name is Dr. Ashwin Ajit. I'm a postdoctoral fellow at Dr. Horuzko's lab at the Georgia Cancer Center.
And my lab, it study two main projects. One is transplantation tolerance, and we look in translational component. Another project is cancer and inflammation. The transplantation tolerance, it's very inspiring because it's provide big benefits for the people who got organ transplantation, tissue transplantation. And my lab is very interesting in molecule which called HLA-G. This molecule involved in natural tolerance mechanism. It's prevented rejection of baby by expression of this molecule in maternal side, and prevented for semi-allergenic baby rejection. So we interesting what is the mechanism for this prevention of rejection. And initially we did a lot of animal model. We used HLA-G and receptor on different cell types. We're looking how interaction between HLA-G and receptor on dendritic cells, on T cells. What is the interaction it's provide downstream, how regulate the function of these cells through the interaction of HLA-G and inhibitory receptor. Right now we are doing a lot of translational research because we're interesting to understand how organ transplant can be prolonged in the patient. And particularly interesting in molecule called HLA-G, and we discovered that HLA-G expression, it's a very high level of expression present in the patient who prolong kidney allograft survival. So we established collaboration with Department of Medicine in nephrology section and transplantation section in Augusta University, and we analyze the level of expression HLA-G, the level of expression of inhibitory receptor on different type, and this is a ongoing research. Some of the part of the research already published, but we also very interesting to introduce new components like we're working on the model called humanized mouse model, and this model is very powerful for the selection of donor. For example, when we've got living related donor, we've got opportunity to select much better the combination between recipient and donor, because this is a very important task. We want to identify best match which potentially will be preventing of allograft rejection, and we study this in creating humanized mouse model with cells from the recipient kidney, and then we doing combination from the donor. We're looking how immune system, which established in humanized mouse model, how this patient immune system can respond to potential donor antigen. We're using a lot of approach, genomic approach, we're using flow cytometry, and so many approach. And then we create algorithm. We can see which combination is more acceptable. And this is a future because the patient doesn't need so many immunosuppressive drug in this combination, and it's prevent possibility of rejection. Another approach which we developed with clinical, it's we want to introduce immune monitoring based on the immune system of the patient, how this immune system it responds to already transplanted organ, because allogeneic rejection it's very often, and we need to understand early stage. So we're looking for very early stage of recognition of transplantation antigen, and we can correct this recognition. If it's very strong immune response, we can adjust with therapy. If it no, we can remove part of personalized medicine. Our transplantation research was currently it's funded by Carlos and Margaret Mason Trust, and we very thankful the Mason Trust for support, because this is give us possibility to bring transplantation research on the high level, where it's a lot of genomic analysis, proteomic analysis involved, and this is a benefit for kidney transplant and another organ transplant patient, especially in Georgia.